Welcome to Miss Lovedoll's video on the cell cycle. This is Stacy Lovedoll. Before we get started, let's check and make sure that everybody has what they need. You need to have your science notebook, a pencil, and some colored pencils. You ready? Okay, let's get started. The objectives of this video are to help you understand the characteristics of prokaryotic cells, to explain the steps in binary fission, and to understand the relationship between DNA and chromosomes. So, humans were made of trillions of cells, and like all living things, we pass through different stages in our life. We grow, we reproduce, we die. Oh, did I leave out we're born? Well, cells pass through different stages of life as well, and we call this the life cycle of the cell. The life cycle of the cell begins when the cell is formed, and it ends when the cell divides and forms two new cells. So that's a little bit different than humans. Our life cycle ends when we die and we are no more. But a cell's life cycle ends when it divides and forms two new cells. And we're going to see how this happens. But first, let's talk a little bit about DNA. A lot of you have heard about DNA, deoxyribonucleic acid. It's the hereditary material that controls the cell. And it has a really unique shape. It's called the double helix. You can see it looks like a ladder that's been twisted. We're going to go into some of the details of DNA in another lesson. The two forms of DNA that we are going to refer to in this lesson are chromatin and chromosomes. Chromatin is uncondensed DNA. So what does that mean? Well, when the DNA is in this form, it's loose and unwound. You can still see that double helix shape, but it's not all coiled up. It's just loose inside the DNA, or inside the nucleus. This is the form that the DNA is in when the cell is not dividing. To me, chromatin reminds me of cooked spaghetti. It's loose and unwound and kind of just in a relaxed state. Kind of looking tasty if we had some sauce. Chromosomes are also made of DNA, but it's DNA and proteins that have condensed the DNA into this very specific shape. When DNA is in this form, it means that the cell is in the process of dividing. And with our spaghetti analogy, it kind of reminds me of uncooked spaghetti. It's got a specific kind of compact shape. So, how are those Cornell notes coming along? Here's an example of the notes that I've taken on the slides to this point. Now, you don't have to copy my notes, but this is just here to give you an idea of what types of things you might be writing down. Press pause if you need to. Before a cell divides, it has to make a copy of its DNA. This ensures that the daughter cells have the exact same DNA as the parent cells. And that makes sense, doesn't it? We wouldn't want our skin cells or our muscle cells or any cells in our body not to have an exact copy of our DNA. Every cell, every time, has to copy the DNA exactly. So how does a cell make more cells? Well, it depends on whether the cell is simple or complex. Simple cells are prokaryotes. That's what we call them, or prokaryotes. And bacteria is an example of a prokaryotic cell. It's very simple. It's a single cell. It's a living thing that can grow and reproduce on its own. But you're going to notice some differences when we compare it to eukaryotes, which are plant and animal cells, which are much more complex. A prokaryotic cell has no membrane-bound organelles. That's one of the key things that makes it a prokaryotic cell. So what that means is that instead of having the DNA inside a nuclear membrane in the nucleus, that a prokaryotic cell doesn't have a nucleus. The DNA just floats around inside the cytoplasm with everything else. It also doesn't have any of the other organelles that we learned about in our cells unit. It doesn't have vacuoles and mitochondria and all those other things that we found in plant and animal cells. In addition, prokaryotic cells have their DNA in a single ring. Now, if you unwound all of this DNA inside this little bacteria cell, you would end up with one big continuous loop of DNA. Let me write that right there. So here's the DNA inside our prokaryotic cell. And that's a lot different than the way the DNA is in a eukaryotic cell, and we'll see that later. So let's do a note check again. How are those notes coming? Press pause if you need to. To make new prokaryotic cells, the prokaryotic cell goes through a process called binary fission. And it's really pretty simple. The DNA is copied, and then the cell splits in two. It produces two daughter cells that have an exact copy of the parent cell DNA. So let's look and see how this happens. The first step is that the parent cell will elongate. That just means that it gets a little bit longer. And the DNA is replicated. That means that this DNA is copied, and this copy is exactly the same as the original copy. Step 2, the cell wall and the plasma membrane begin to divide. Now, don't get confused by plasma membrane. That's exactly the same as cell membrane. Remember, in science, a lot of times there are different words for the exact same thing. So cell membrane, plasma membrane, same thing. Then, step 3, a cross wall forms completely around the divided DNA. So we can see here's our cell wall, and now it's formed and completely enclosed the DNA on one side and the copy on the other side. And then step 4 is the cells separate into two new cells. These two cells have the exact same DNA as the parent cell. Over here we can see a copy of a bacillus, which is a type of bacteria that is in the process of dividing. So, how are those Cornell notes coming along? Press pause if you need to. Binary cell division is fast. Let's look at how fast bacteria can reproduce using binary cell division. So, let's look over here. Suppose a bacteria divides every 20 minutes. So we're going to start off with one little bacteria cell. I'm going to make them round just for the sake of making it a little bit easier. So after 20 minutes, if this cell divides, it's going to turn into two daughter cells, right? So that makes sense. 20 minutes, we've got two cells. Okay? Now, let's take a look and see how this very quickly gets out of control. What's going to happen after 40 minutes? So we start here with one cell. 20 minutes later, we end up with two cells. Okay, so we're going to put over here 20 min. There we go. And let's see, another 20 minutes is going to go by. And after 20 minutes, we will have, this cell will divide in two. So we'll have two cells from this parent. And this cell is going to divide in two. Now you notice my cells are getting smaller just because I'm going to start running out of room. So 20 minutes, we get two cells. Another 20 minutes goes by. And how many cells do we have? Well, 4, right? 1, 2, 3, 4 daughter cells after just 40 minutes. Okay, does that make sense? I hope so. Let's go to the next slide. How many cells would there be after 80 minutes? So here we have 20 minutes for every cell division, and we're going to let 80 minutes go by. So first question is, how many cell divisions are there in 80 minutes? Well, we would take 80 and divide it by 20. Okay, and what does that give us? Well, we can cross out those zeros. 8 divided by 2 equals 4. So this cell is going to divide 4 times. So let's take a look and see how that happens. Now you should be working this out on the opposite page of your notes. So we start off with our single cell. Okay, does that make sense? I hope so. Now let's step it up a notch and see what happens after 80 minutes. Suppose we have our same bacteria and it's going to divide every 20 minutes. How many cells would there be after 80 minutes? Work out your answer on the opposite page. While you're starting on that, I'm going to get myself a blank slide. Okay, so here we are with our blank slide. We have 80 minutes, and we want to know, first of all, how many cell divisions is that? Well, if we have 80 minutes to wait, and we have 20 minutes for every cell division, we can take 80 divided by 20. Let's cross out those zeros, and that gives us 8 divided by 2, which is 4. So in this case, the cell is going to divide 4 times. Okay, so we'll write that like that. So here's our parent cell. And after 20 minutes, that parent cell is going to divide in 2 and form 2 daughter cells. So let's put those over here. I'm going to try to do this neatly so you guys can see it. Now, 20 minutes is going to go by again. So this is our second cell division. And this one is going to divide in 2 and produce 2 daughter cells. And this one is going to divide in 2 and produce 2 daughter cells. So there we go. So now we've got 4. We know that because we've done this before. Okay, so here's our third cell division. Another 20 minutes goes by, and these 4 cells all divide. This one divides in 2 and produces 2 daughters. This one divides in 2, produces 2 daughters. This one divides in 2 and produces 2 daughters. And this one divides in 2 and produces 2 daughters. So now we have 1, 2, 3, 4, 5, 6, 7, 8. But we're not done. We need one more round of cell division. So here's our next 20 minutes goes by, and all of these divide in 2. So this one divides in 2 and produces 2 daughters. Now, if we can count these all up, how many do we have at the end of 80 minutes? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So after 80 minutes, we end up with 16 cells, where we only started with 1. Okay? So we have 2, and then we have 4, and then we have 8, and then we have 16. So you can see that after every cell division, when you're doing binary fission, you end up with twice as many cells. So the first cell division, we ended up with 2. And then after the second cell division, we ended up with 4. After the third cell division, we ended with 8. And after the fourth cell division, we ended with 16. So here's a question for you. How many cells would we have after 6 cell divisions? What do you think? Write your answer in your notes, and we'll go over it tomorrow when we get back to class. So this brings us to the end of the first part of our cell cycle series, where you've learned about prokaryotic cells and how they produce more cells. Don't forget your 3 R's. On the opposite side of each page of your notes, do some kind of a reflection, 30-word summary, an illustration, or a diagram, and respond to these questions at the end of your notes. Number 1, why is it important for DNA to be copied before cell division? And number 2, what are 2 ways the prokaryotic cells are less complex